Hi, my name is Jonathan Fallowfield and I'm a liver doctor and a research professor at the University of Edinburgh. You probably don't know that January is actually Love Your Liver Awareness Month. This is a campaign run by the British Liver Trust, and I strongly encourage you to visit their website to learn more about the campaign, find out ways you can love your liver more over the next month, and also ways to fundraise for this excellent cause. We'd also like to take this opportunity to tell you about some of the exciting liver research that's happening in Edinburgh. This ranges from studies in the laboratory through to clinical trials.
Hi there, my name's Neil Henderson. I'm a liver consultant and liver researcher based in Edinburgh. So the liver's an incredible organ. Without a liver, we can't survive, and it's incredibly important in lots of different ways. It performs thousands and thousands of functions for us. It allows us to absorb the nutrients from the food we eat and build them into proteins and other important molecules that are so important for our health. And it also is a major production site for bile, which we need to absorb all the goodies again from what we eat. The other very important thing is it detoxifies our bodies, and that's one of the major functions of our liver. So for these reasons and many, many others, the liver is an absolutely vital organ.
Hi, I'm Tom Bird. I'm an NHS consultant and I also lead a research group at the CR UK's Beatson Institute. I'd like to tell you a bit about what we are doing to tackle liver cancer. Firstly, we're aiming to improve the early detection of this tumour. This is important because any cancer detected early stands the best chance of being cured. And to do this, we're combining improvements in tumour markers within the blood with advanced medical scanning and state-of-the-art mathematical analysis. And this will allow us to identify people most at risk of cancer, but also improve the way we perform cancer screening within the NHS. The second area we really want to make a difference is in precision medicine in liver cancer treatment. And if we understood every individual cancer in every patient in as much detail as possible, then we could unlock what makes that cancer tick and find the right drug to treat that cancer. And together we're making exciting strides towards giving every patient with this disease the best chance of curing their tumours.
Hi there, my name's Neil Henderson. I'm a liver consultant and liver researcher based in Edinburgh. So in Edinburgh, our research over the last few years has used a cutting-edge technology called single-cell genomics. Now, the reason that we're excited about that is because previously we could take a piece of human liver and measure all the genes within that chunk of liver tissue, but we'd really no idea where each gene came from within the liver. But with single-cell genomics, this is what we like to call a resolution revolution. It's like going from a black and white TV to an ultra high definition colour picture now, and it's really an incredible step change in our ability to look at gene expression within the liver. So we can do this at single cell resolution now, and so what we're excited about is using these new technologies to really zero in on these bad cells and inhibit scarring in the liver. And ultimately we hope that this will negate and stop the need for us to do liver transplantation.
Hi, I'm Tim Kendall. I'm a pathologist, which means I use a microscope behind me there to look at glass slides, make a diagnosis in a clinical setting, or give some descriptor of research value. But what we really want to do is now use computers to get more information from stained sections than we can do by just looking at those ourselves. This section here has been stained for scarring in the liver. Scarring is red, the white hole is fat, and the yellow is just the background liver. And we can train a computer to identify the scarring and the fat and the liver and classify that into a different colour so it can count the number of pixels. As we see here, and that allows us to get a more accurate assessment of features that we as pathologists are unable to do. And when you put the two together, the human assessment and the computer assessment, you get greater value.
Hello, my name's Ruth Andrew. I'm an analytical chemist, which means that I use analytical technologies to help understand the composition of biological samples. So, in other words, what is the liver made of? We use sampling devices such as lasers to take very, very small samples from biopsies that come from our liver doctor colleagues, and we then list out the molecules that are within those samples. We can do that in healthy livers, we can do that in diseased livers, and hopefully by understanding how these molecules change, we can have some chance of finding ways to intervene in early disease and identify patients that are at risk of developing disease. We don't do this just in one sample. We take multiple samples from the biopsy, and actually we use that all to reconstruct a map of the liver. We can do this in 2D or 3D, but ultimately we can see the landscape of the liver.
Hello again. So one of the subjects I'm interested in researching is something called non-alcoholic fatty liver disease, or NAFLD. And NAFLD's really important because it's the commonest cause of chronic liver disease worldwide. It affects a staggering 1 in 4 adults. But critically, not everyone with a fatty liver goes on to develop severe liver problems. In fact, out of 100 patients with fatty liver, only around 5 will develop end-stage liver scarring, or what we call liver cirrhosis. We really need to understand this. We've got no good tests for the condition, and we have no medicines available to treat the condition. To tackle this, we've set up a project called Steatocyte, a collection of around 1,000 fatty liver samples of varying severity drawn from across Scotland. We want to analyse these samples to identify the genes, the cells and the molecules that are driving bad liver outcomes in the small proportion of patients that are badly affected. We hope to improve the management of this condition.
Hi everyone, I'm Maria. I'm a second year PhD student in the CIR, and for Love Your Liver Month, I would like to tell you a little bit about my PhD project. I am using the clinical and RNA-seq data from Steatocyte to find different gene signatures for the different stages of non-alcoholic fatty liver disease. With this, we would like to identify potential combination therapies in silico and then test them with microfluidics by measuring the expression of key genes that participate in the progression of the disease, with 3D cellular models, and then ex vivo with precision human cut liver slices. So ideally, by the end of the PhD, I would have combination therapies that could be a possible treatment for the disease.